Okay, so the end of mapping here. So you thought that dihybrid cross was tough. They do trihybrid crosses, quadrid hybrid crosses. I mean, it's crazy the number of traits that they'll track at the same time. In this case, they're looking at 3 traits. They're looking at, here are the 2 parental flags, the female and the male. In this particular case, the male is the dominant, or the wild type. And they do a test cross. They'll do a series of test crosses. I am just going to go through this a little bit because this is, showing something called a double crossing over. And so in this particular case, what they're doing is they're looking at a number of sites, these 3 sites, see how far they are apart. And for one of them, gray body, red eyes, black body, purple eyes. Yes, flies can have purple eyes. They had, 944 individuals of the parental offspring, and what that means is that 944 look like the parents did. The non-parental here is another term for the recombinants, and so when you calculate the map distances, you can see that eye color and body color are 6.1 map units apart from one another. They did this for a number of different traits: body color, wing shape, eye color, wing shape, and on and on and on and on. And so here are 3 other traits, VG, PR, and B, and they were able to figure out or construct an individual map showing the distance among these 3 genes. Now, the sum of the distance between body color, eye color, and wing shape, it turns out, is actually greater than that between body color and wing shape. What? So you have 18.4, which is, body color, eye color, wing shape, versus 17.8. Why is that the case? It's because what you're doing is you're looking at a statistical average. When you're looking at and counting recombination frequencies, what you're dealing with is the number of animals that you got with those phenotypes at that point in time. Sometimes you'll get a few more of one type than another type, and so that's going to move that map distance around just a little bit. Another issue comes out when you have something called double crossing overs. Double crossing overs happens when you have enough time for the event to occur twice, or you have 2 chiasmas forming on 2 adjacent sister chromatid pairs. When that happens, then let me just scroll through here until I get to the pictures. This is all the math showing this. The double crossing over. Where's my picture? This is what I wanted to show you. A double crossing over event is going to mix up, say for example, instead of having these 2 tips, SN and Y swapping completely, what you'll have is SN swapping, but not Y. So this Y would be over here, and this Y would be over here. So sorry, I had to scroll through a lot of pictures there to show you kind of what a double crossing over looks like. In fact, why don't I just draw it here? So you have a pair of sister chromatids like this, and another pair like this. And we know that a crossing over happens when just the tips touch each other, right? But what will happen is you'll have one tip and another tip touching here and touching here, so that only this intermediate region swaps from one side to another. It's kind of crazy when you see it. The inferences and the product rule. Since we didn't follow this, and since in this particular course the outcomes don't require that we do a whole lot of statistics, basically we'll just review this real quick. The probability of a double crossing over is the same as or equals the probability of a crossing over between B and PR and VG. In other words, you're looking at not just the probability of a single crossing over, but 2 crossing overs happening at the same time. And because this is the product rule, you have to multiply the probabilities by each other, which tells you that you have 0.0075 chance. In other words, you have like a 0.75% chance. Or let's do it this way. This might make more sense. 0.75% chance of a double crossing over to occur. Actually, it's 7.5. I'll straighten out my math there. So you have to have a lot of offspring to see this, and that's why they're showing the mathematics with all these different groups. So let's finish up here. So crossing over occasionally does occur during mitosis. This is kind of a weird thing. Occasionally, very occasionally, a crossing over can happen during mitosis in cells, in somatic cells. And so what that means is that if for whatever reason during mitosis you have a crossing over event happen, then you're going to have what's called mitotic recombination. In other words, the new cells being produced from that single cell division is going to have a new combination of alleles. It's going to express things differently. So it may actually have a different phenotype, like a different pigmentation. If this happens during early development, the daughter cells containing those chromosomes will continue to divide and continue to reflect that recombination event that happened mitotically. So it turns out they saw this in Drosophila, and this is another scientist named Stern, Dr. Stern. He was working on strains of Drosophila that carried X-linked genes affecting both body color and bristle morphology, or bristle shape. And so you have short bristles right here, if I can get it to highlight short body bristles, or normal body bristles. Okay, I don't know why this won't highlight short. Sorry about that. And then you have the body color. So when he looked at the flies, he noticed that along the body of the fly, sometimes you would see short bristles, and sometimes you would see long bristles. And this happened in different patches of the body. So if you look at the side of the Drosophila there, for example, let's say you look at just this patch right here. This patch would have little tiny short bristles. But then when you go and look at a different patch on the surface of the animal, it had really long hairs, really long bristles. It's kind of weird. It's kind of like, it's not the same thing as X inactivation. This was just a consequence of a crossing over event that happened during mitosis. And as it turns out, every single one of these cells, or every one of these hairs, comes from a series of cells that make long bristles. Over in this other region, each one of these hairs comes from a set of cells that makes short bristles. And so you have a clone. This would be the short bristle clone. And over here, this would be the long bristle clone, right? So in that particular case, what you're doing is you're tracking a set of cells as they divide over the lifetime of the animal, and each set is considered a clone. Kind of cool, right? Kind of like that. All right. This is the end of gene mapping. Really interesting topic. It turns out that the maps, the genetic maps that Morgan and Sturdivant developed in the early 1900s ended up being almost on the spot when geneticists sequenced the Drosophila genome. There were only a couple cases in which, and I'm going to draw here on the side, in which, let's say along the length of the X chromosome, you're looking at individual genes. There were only a couple cases where they were inverted or swapped. And why do you think that is? And how often did you see that swapping occur? So let's say this gene and this gene, they were swapped with one another, all right? In the original map that was made in the early 1900s versus actually sequencing the genome. You'll never see a swap between genes that are far apart, though. And the reason is because the recombination frequencies were so low. Let's say this was a 2% map distance, or Morgan unit, MU, right? Let's get rid of that. Let's make it a Morgan unit, or map unit. A 2% map unit happened because the probability of getting that recombination event was so small that it was somewhere, you know, it was like, okay, if this was A and this was X, they might have mapped A above X, but it turned out that it was really X above A. But since there was only a 2% difference between the two, it was easy to make that mistake. If this was P and this is Q down here, and you had like a 70% difference, okay, or 70% Morgan units, or 70 Morgan units between the two map units, then you wouldn't really mess that up. You knew that P was at the top and Q was at the bottom. All right, so we got to use the pen today. It worked okay. I'm going to keep working on this. Maybe I need to, I'm playing with different sizes of tips and stuff. Maybe a slightly thinner tip would work better. I don't know. You guys tell me what you think. All right, I am going to sign off for now, and I will see you in Chapter 9. Remember, we're skipping Chapter 8.